So this is the second part for Chapter 4, Innate Immunity. And so we'll be talking about some of the processes that go on, so cellular processes that go on in the innate response. So the first audio lecture for this chapter, we talked about different receptors and also the physical and chemical barriers. So the first process that we're going to look at is looking at phagocytosis. So I've mentioned this term numerous times, and so finally we're getting to it. So with phagocytosis, this is defined as engulfment and internalization of matter such as microbes for their clearance and destruction. And so phagocytosis is where an immune cell will engulf a foreign cell or dead, damaged cell. Okay, and the way in which this happens is a multi-step process. And so the first step is where we need to get binding of receptors that are on the immune cell to ligands that are on the microbe or damaged cell. So this is where our pattern recognition receptors, PRRs, and our MAMPs or DAMPs will bind. So what happens is these pseudopodium come out because we have this rearrangement of the actin microfilaments. So this is like cell bio 101 coming back. So when you talked about in cell bio about the cytoskeleton, about actin microfilaments, and how you can get elongation and that can change the shape of the cell, here you go. So that pseudopodium are created by this rearrangement of the actin so that it will actually envelop the microbe or the cell that is being engulfed. Okay, so that material, when the pseudopodium closes around it, and I think of it as like a bear hug, it ends up putting it into a vesicle, and that vesicle is going to be the phagosome. So the phagosome is going to contain the bacteria or foreign material. And so in the figure, you can see how the pH is, kind of around a neutral pH. And then right to the left of our phagosome, we actually have a lysosome. In that lysosome, you see like little purple kind of dots. That's the digestive enzyme. So what's going to happen is the phagosome and the lysosome are going to fuse together. When that happens, there's a drop in the pH. That lower pH, bacteria doesn't like that. The bacteria gets killed, and plus with the digestive enzymes, it gets digested down. Okay, and so then we can see here in the later stage, this phagolysosome now has the pathogen, but it's all dissolved up and broken into little pieces. So that material, that digested product, can be released from the cell through exocytosis. That is going to allow for that material to be moved out into the extracellular fluid around the cell. But we can also take some of that material and put it on receptors that can talk to other immune cells. So just keep that in mind. This process of phagocytosis allows for these innate immune cells to interact with adaptive so that they can show antigen. So the initiation of phagocytosis is through the recognition of these PAMPs, these MAMPs, DAMPs, kind of this non-self, right? Kind of a generic response that's classic of the innate system. But in the end, the presentation can be of specific antigen. Okay, so, and it's just that all these different products that are formed are put on receptors. It's not that this immune cell says, oh, I know that this is E. coli flagella. I'm going to put it on a receptor to show it to T cells that know this. It just says, oh, look, here's all the stuff I ate and little pieces of it. And then it's checking to see whether, for the adaptive response, whether the T cells and B cells recognize it. And we'll talk more about that in our next unit when we talk about adaptive. So those receptors we had talked about earlier, those pattern recognition receptors, can trigger phagocytosis. As I mentioned with the C-lectin receptors, this is where we can get recognition of the lectin and amylose that is found on microbes, and we can also then get activation of complement, which we'll talk about later on. There's also scavenger receptors that are able to recognize LPS and other molecules that are coming from microbes. So this is just looking at the mannose binding lectin receptor, and then we also have this C1 complement protein 1 binding to LPS. So we can get this recognition. So these can be these receptors that allow for the recognition to cause phagocytosis. And we'll talk more about these the binding that's occurring when we talk about complement. So I put this in to kind of foreshadow. So as I mentioned, the material that's taken in gets put into a phagosome. So again, another term to keep to remember. Phagosome is a vesicle that contains the pathogen that is then going to fuse with the lysosome. Lysosomes where our digestive enzymes are, and so it's through enzyme degradation, also the production of antimicrobial peptides, and then we also have a lot of reactive oxygen species and reactive nitrogen species that are present in that phagolysosome, so that allows for the pathogen to be destroyed. So this figure in the textbook just shows you how reactive oxygen species and reactive nitrogen species are formed from oxygen and nitrogen. Again, it's just, if this is kind of a passion of yours is understanding this, then go for it. It's not that you're expected to memorize this. So next we'll look at inflammation. So inflammation is going to be initiated by pro-inflammatory cytokines and chemokines. So if you remember to part 1, we had talked about how when these cells recognize when the receptors and ligands bind, one of the downstream effects is production of cytokines. These cytokines and chemokines will then initiate a response that we would call inflammation. Okay. With inflammation, you're going to get increased vascular permeability. So blood vessels are going to open up, and this allows for immune cells that are in circulation in the blood to actually move into the areas of tissues. Okay, so it's going to cause like a leakiness of the endothelial cells, and we'll look at this in the next figure so you can see it. Because of this leakiness, it allows for recruitment of neutrophils and other leukocytes from the blood into the area of damage. Okay, so this is a good figure. Also, that phagocytosis figure is another good figure from this chapter. So this figure outlines inflammation and the steps that are happening in order to have inflammatory response. Okay, so again, worth printing out. So for inflammation, we're going to have some kind of tissue damage. That tissue damage may lead to entry of bacteria, but this is going to cause some of these resident cells to respond. So in our skin, our resident cells would be resident macrophages. We'd also have mast cells, and so you might remember mast cells release histamine. So they're going to end up degranulating, releasing histamine. That histamine is going to cause vasodilation. So there's a response from the vascular system because of that. The histamine also can act as a chemical attractant to recruit in cells. So it's kind of like a double whammy. We have other factors that are being produced by these damaged cells that also initiate and help in this process. Okay. So that's all like step 1. So then looking at step 2, what happens in response to histamine and other components is that we get changes in that vascular system in the capillary. And so you'll notice that there's these pancake cells. They look like pancakes in here. Those are the endothelial cells that are going to line the blood vessels. So when we get vasodilation, it actually opens up the blood vessel and makes it bigger. That allows for more blood flow through the area, which means we're going to get more cells through the area, right? So more blood flow, bring more cells, potential immune cells into the location. That vasodilation also causes some separation between those endothelial cells. So just think about, like, if we expand something, there's only so much that the cell can stretch. So we end up with some leakiness or permeability of the capillaries. So this is going to allow for those immune cells to squeeze on through. And so this process is commonly called as diapoesis, but nowadays it's called extravasation. I'm not sold on it being called extravasation, because I think of that as a process that cancer cells undergo, and diapoesis is a kind of more general term, and I understand it's just because I have gray hairs, so it's just an updating of the terminology. I'm just not quite there yet. So I accept either one when it comes to exams, just because if you're looking at older textbooks, they're going to not old, but just even a couple editions, even like 5 years ago when I was teaching, it was diapoesis, and also we made this change, and I'm like, what? And I had done some cancer research, so I was like, these are not cancer cells, these are immune cells. So you can see I'm having some issues with it, but I'll get over it. Okay, so this allows for those neutrophils and other immune cells to move into this location where they can then help in undergoing the process of phagocytosis with material that's there, and whether that is bacteria, pathogens, or whether that's damaged tissue. Okay. They're additionally producing cytokines to cause chemotaxis, to draw other immune cells in there. So we're kind of renewing the signal for, come here, something's wrong, come help us, okay? And I do apologize that this is a virtual lecture. Normally when I'm in a classroom, I use my classroom as the site of damage. And then our hallway is our blood vessel, and I squeeze through the doorway, which is our endothelial cells separating. And so you have this really nice visual. But hopefully talking you through this helps. And maybe when we do our, video meeting, I can, put the camera on and I can do my normal little dance routine. Okay. So when we look at inflammation, there are later stages of inflammation, and these are known as the acute phase response, or APR. And so this is induced by pro-inflammatory cytokines. So again, we want the response to continue so we get repair, and so cytokines are going to help to make sure that happens. With APR, acute phase response, we're going to get increased synthesis and secretion of antimicrobial proteins, because as that repair happens, we want to make sure that we don't have new pathogens coming into the area, or maybe if we didn't clear a bacteria, we want to make sure that we have a response to it. So there's a number of different proteins that are involved in that are produced by the liver. So the liver acute phase proteins get activated, and they're going to help to eliminate those pathogens. So just to kind of, complete the story, we want to look at some of these other immune cells. And so we have natural killer cells, NK cells. So these are actually lymphocytes. So they're going to be derived from the progenitor, lymphatic progenitor cell line. But they're going to have innate functions in that it's a recognition, not so much of self versus non-self, but damage, okay? So they can respond to infections, malignant transformations, and other stressors. So they're able to sense whether a cell, your own cell, has been damaged in some way. And so an activated natural killer cell can kill altered self cells. And they do this by releasing porphyrin. porphyrin causes little holes in a cell. We'll see a similar structure later on when we talk about complement, but they cause these little pores to be created in the hole, so their insides leak out. And then also granzymes, and those are enzymes that will actually induce apoptosis. Activated natural killer cells can also produce cytokines that actually recruit in the adaptive response to then target these altered self cells. So natural killer cells are widely studied in cancer because they normally take care of altered self, so cancer cells, but cancer cells have a way of kind of tricking them sometimes and not responding. So for aggressive forms of cancer, it's an area of research to see what kind of role they have and whether there's a way to kind of reprogram the natural killer cells to be able to see these altered these cancer cells again. And then we have our innate lymphoid cells, ILCs. So again, these are also derived from our common lymphoid progenitor cells. They do include the natural killer cells, but there's also other populations. So usually natural killer cells kind of get their own thing, and then the other ones, the poor other ones, are left as this miscellaneous category. Okay, so generally, if you're looking at natural killer cells, you will say natural killer cells, and then the other groups end up being called innate lymphoid cells. Okay, each group has different cytokines profiles that they produce. And our innate lymphoid cells, they lack the pattern recognition receptors, so it's not that they're being activated directly through pathogens. There's this link to this YouTube video that does a really nice job of explaining what innate lymphoid cells are and what their role is. So I'll be honest, when I was an undergrad 20-ish years ago, there was no innate lymphoid cells. Natural killer cells were like a thing potentially, but again, so this is a kind of newer cell type that's been identified, and again, it's through its cytokine profile, what cytokines it's producing, and then receptors on its surface that we recognize that there's these subsets that have a slightly different function. So this is just looking at 3 of the different groups, so they can be grouped, and then the different populations. So there's NK cells, and then there is innate lymphoid cells 1, 2, there's LT1, and then there's 17 and 22. Okay, so are different groups. And the way they're generally grouped is by their function. So our group 2, or our innate lymphoid cell 2, they're going to provide immunity against hemolymph or parasitic worms, and also with wound healing. And again, the cytokine profile would be unique to those. So next we're going to look at some of the regulation and evasion of innate and inflammation response. So keep in mind, if you're a pathogen, you want to have strategies in order to block or evade these responses. But as us, the host, we also want ways in order to regulate that. And so at us as the host, we could have defects in our pattern recognition receptors or the signaling pathways, and that would lead us to be more susceptible to infections, right? So someone who has a mutation to a TLR wouldn't be able to respond to those pathogens specific to that TLR as well. And we can see this if we have mouse models that have mutations. They're going to be more prone to infection, but then it's obviously there's going to be humans that are going to have mutations or defects in these receptors and also their pathways. So in some cases, this may actually lead to the system to be turned on. So normally there's going to be on-off regulation, and so if these defects allow for the system, the response to be in an on position, this is going to cause inflammation, chronic inflammation, and so we can get, that's obviously not a good thing either. So, type 1 to type 2 diabetes, part of the issue with wound healing is that they're under chronic inflammation. Okay. And so with regulation, there's going to be both positive and negative mechanisms in allowing that to happen. And again, pathogens are going to have ways to get around and evade these responses. And so the table we're going to look at next looks at some of these different strategies. So table 4-7 looks at some of the strategies to evade. So they can try to avoid detection by the pattern recognition receptors. And so this is where they might, a mutation or a change to a protein actually will get selected for because it doesn't get recognized. Okay, so again, we want to be mindful. It's not that the microbe says, oh geez, let me change that. Mutations happen, and then those ones are the ones that don't get cleared, so they get selected for. Okay, so viral proteins can change, and again, they would be selected for. We can also block pattern recognition receptor signaling pathways, and that's going to prevent activation of the response. And then we can also prevent killing or the inhibition of replication. And so this one's probably the most widely understood because the process of phagocytosis is well studied. And so this can be where a microbe can produce a neutralizing component against the digestive enzymes that are going to be produced by the lysosome, or it could prevent the fusion of the lysosome and the phagosome, so we don't get that. And so some of these microbes could actually live inside the cells, which is kind of pretty cool. And then other ones will actually produce leukocidins, will produce chemicals that actually kill the immune cells. So lots of different ways in which a pathogen can still survive and kind of outsmart at least the innate response to begin with. So there's several innate systems that have been co-opted by the adaptive to contribute to antibody-mediated pathogen elimination. And so what this means is that there's an overlap between innate and adaptive. So if the adaptive B cells are going to produce antibodies, those antibodies can be utilized in the innate system, and this is the processes of opsonization and complement activation. Okay, and so there's again this idea of this kind of overlap, and then also some lymphocytes are also able to express TLRs, and they actually use them as co-stimulatory receptors. So you have your main, like your T cell receptor and MHC interacting. But then you also have to have these co-stimulatory receptors, and so TLRs can act as that. So there's kind of, overlap in the systems. So dendritic cells become a key bridge between innate and adaptive because dendritic cells are really good at picking up through phagocytosis antigen and then bringing it to T cells in the lymph nodes. So dendritic cells will traffic from the site of infection to a lymph node, and then they will interact with the T cell. And so what we're seeing in this figure is our dendritic cell, and then it's going to take antigen, material that it's undergone through the process of phagocytosis, and present it to these different cells that are in the lymph node. And so if they see this, if the antigen that's being presented is the antigen that the receptor binds, we'll have these naive T cells becoming mature T cells and differentiating into the different subsets. So we have some T helper cells, cytotoxic T cells, and then we also have the T regulatory cells and additional T cells, and also B cells could become activated. So we want to keep in mind that innate immunity is evolutionarily older than the adaptive. So, we are born with our innate immunity in which our parents gave us, whose parents gave it to them, whose parents gave it to them, where our adaptive immune response is different, right? It's personalized based on what we have seen, and so there are parts of the innate system that are conserved all the way through to plants and such, and we'll look at this table next. The next table, looking at some of these different multicellular organisms and what components they have, but something as basic as that leucine-rich repeats that's found on the pattern recognition receptors is found on all plants and all mammals. The TLRs themselves are unique to mammals, so there's a slight variation, but again, the kind of basic structure is conserved. So this just looks at some of the different components, starting at the left, looking at innate immunity, then adaptive, and so you can see how in different, we'll say, lower level organisms like invertebrates and earthworms and arthropods, they do have an innate response, but they lack an adaptive response. Okay, so again, these organisms have cells that can undergo phagocytosis. They produce antimicrobial peptides. They have pattern recognition receptors. But then as we move to the right side of this chart, when we look at adaptive things like lymphocytes and our variable lymphocyte receptors in antibodies, these lack them. So the adaptive immune response is really unique to vertebrates. So again, just keeping this in mind some of us love evolutionary biology, and so just to keep in mind that we can look at the immune response in organisms, and we can actually see how it has evolved to higher order organisms. And I don't really like the term lower order and higher order, but thinking of mammals as being more complex, and I don't like that term either, because a simple bacteria is very complex. But we have multiple organ systems and such, and so we have more of these features. So to summarize this chapter, we want to keep in mind that we have basic barrier mechanisms, both physical and chemical, that allow to be the frontline defense of immunity. And so that is our first line. And so when we have a breakdown in our first line in these barriers, physical and chemical, that's when pathogens can be introduced. Potential pathogens, microbes can be introduced to us, and it's that innate immune response that helps to have a general recognition of pathogenic molecules, and that the response could be phagocytosis, it could trigger inflammation, we could have direct destruction through the natural killer cells, and then initiation of adaptive immune response. Okay, and we can have all of these responses, so it's not a one or the other. All of these responses can occur when we have activation of the innate response. And as someone who really focused on adaptive immunity, it's really important to understand innate immunity because it's going to set up the adaptive response, and without a good innate response, you can't get a good adaptive response. And so having that understanding in the foundation of the cells, of the components, of the processes that are happening in innate really makes it easier, or relatively easier, to understand adaptive processes that are occurring. Okay, so as always, if you have any questions regarding any of the information, either this part or the first part, please do not hesitate to send me an email. If there's any figures that you would like me to go over in more detail during class, I can, or even on one-to-one. All right, thank you very much.